Hi dear students, welcome back to the second session on virulence factors. Previously we had mentioned about the virulence factors such as addition factors, colonization factors, as well as those factors which help the microorganism to invade from one tissue to the other. Now we are moving on to the other virulence factors which would contribute to the virulence of a pathogen. Toxigenicity plays an important role in the case of microbes. In the form of exotoxins or endotoxins, the presence of toxins which have been produced by these organisms aid in their virulence. You know what is virulence? Virulence is the ability of the organism to cause a disease, and a quantitative measure of that is referred to as virulence. If you go to look into the toxins, there are mainly two types of toxins, exotoxins as well as endotoxins. Exotoxins are being found to be extracellular in nature, and they are secreted by the cells and they diffuse into the surrounding media. But endotoxins, as the term implies, they form a part of the cell wall and they do not diffuse into the surrounding media. And exotoxins, they are proteinaceous in nature and they are heat labile. That is, by heating, the toxigenicity or the toxic nature of that particular toxin will be lost on heating. On the other hand, we have the endotoxins, which are lipopolysaccharides, and they are being found to be heat stable. And if you go to look onto exotoxins, they are readily separable from cultures by physical means such as filtration. But endotoxins, since they are intracellular, you need to lyse a cell in order to obtain an endotoxin. Exotoxins often have enzymatic actions and endotoxins do not have enzymatic actions. And you can see that specific pharmacological actions have been found to be associated with each and every exotoxin, and they have specific tissue affinities when we consider the exotoxins. But in the case of endotoxins, they are not being found to be very much specific, and the action of it is somewhat similar to there is a mode of action of these endotoxins are somewhat found to be similar. And exotoxins are active in very minute doses, and they are being found to be highly antigenic. You know what does antigenic means? Antigenic is a substance. If a substance has been found to be antigenic, it triggers the immune system. Okay, so if a exotoxin happens to enter into your body, it serves as a very good antigen and it triggers an immune response within you. But in the case of endotoxins, they are active only when large doses of an endotoxin has been present, and they are being found to be weakly antigenic. And the actions of exotoxins are specifically neutralized by antibody, but neutralization by antibody is being found to be ineffective in the case of endotoxins. Now, when we told about exotoxins and endotoxins, these are just general characteristics how we distinguish between them. Now, what are the examples of exotoxins as well as endotoxins? If you go to look into the exotoxins, a wide range of organisms can produce exotoxins. Usually, gram-positive organisms predominantly produce exotoxins. Now, the most common members are like you have Clostridium botulinum, that's an organism which causes botulism, and the toxin which it produces is a botulinum toxin. And you can see that the gene which has been coding for this particular toxin is being found to be associated with the prophages. And another one is Clostridium tetani, which causes tetanospasmin. Okay, it is encoded by a plasmid. And in addition to gram-positive ones, you can also see various gram-negative organisms. Okay, there are different other gram-positive organisms also which produce exotoxins, but I just mentioned about two among them which are being found to be prominent. And you can see here in the case of diphtheria also, which Corynebacterium diphtheria, the toxin has been produced by a phage which has been associated with it. Okay, so Either the plasmid, the chromosome, or the prophage or the phage associated with the bacteria could contribute to its toxicity. And these toxins which have been coded now, which I have enlisted here, they are being found to be what? They are being found to be exotoxic in nature. That is, they are being produced extracellular or outside of the particular cell. And you can see that gram-negative organisms also do produce exotoxins, and most predominant among them is the Shiga toxin, which has been produced by Shigella dysentery, and it is chromosomally encoded. And E. coli or E. coli O157417, sorry, O157 Het7, okay, that's the variant, the pathogenic variant that which we have. It produces a endotoxin, Shiga-like toxin, and it has been found that it can either be located by the plasmids, the phage genes, which have been integrated into the chromosome also. And in the case of cholera also, the Vibrio cholera produces a cholera toxin, which has been found to be phage encoded. If you If we analyze exotoxins, you can see that even in nanogram concentrations of, that is nanogram per kg of body weight, even if in that particular concentration exotoxin has been present, it has been found to be what? Lethal in nature. Most of them are being found to be lethal when you consider them. Okay, so even in such small concentrations, if you get a what exotoxin within your body, they could be lethal to your body. So that's the reason why botulinum and all that is botulinum toxin, which causes botulism, is being found to be lethal in some cases. And you can see that in several cases, these exotoxins also do serve as super antigens by mainly stimulating 30% of the host T cells to overexpress and express massive amounts of cytokines and from the host immune cells in the absence from other hosts in the absence of a specific antigen. So what you have to look is, see, we have two different immune responses, either a antibody mediated response as well as a cell mediated immune response. In the case of exotoxins, they are so when an antigen comes, normally either the B cell response is triggered. B cell response is the one which produces antibodies, and the second one is the T cell response where the T lymphocytes gets activated. In the case of exotoxins, these mainly serve as super antigens. Okay, super antigen is something which would we would be talking in detail in the case of in your immunological sessions. But this antigen, it mainly activates the T cells non-specifically, and as a result, large amount of cytokines are being produced from the immune cells. And so these excessive cytokines, they cause multiple host organs to fail and giving the pathogen a time to disseminate. So in fact, what would happen when the antigen, when the exotoxin produced by the microorganism is being handled over here, the pathogen is getting what? The presence of of these exotoxin induces the T cell response and large amount of cytokines. These are all chemokine-like substances which has immunoregulatory role. So when lots of cytokines are being formed, what happens? The multiple organs of the host is get targeted. And at the same time, the microorganism which is producing these exotoxins will also get time to disseminate or spread throughout the body. And the best example of a exotoxin is the staphylococcal enterotoxin B, which exhibits a enterotoxin like it mainly has a super antigen activity even at nanogram concentrations. And it is so potent that it is classified as a selected agent because it has the potential to be misused as a bioterror agent also. So though we consider these exotoxins to be extracellular in nature, their role in some microbes come to be very much significant because they could serve as super antigens. Now, this is a schematic representation where the next one, the endotoxin works. If you go to consider the endotoxins, okay, we have already spoken about the exotoxins. Now let's move on to the endotoxins. The most importantly present endotoxin is the lipopolysaccharide. The lipopolysaccharide, I guess you recollect it that it is a part of the outer membrane of the gram-negative organisms. These endotoxins, they mainly indirectly induce a fever in the host by causing macrophages to release endogenous pyrogens, and pyrogens are something which causes fever. Okay, and one among of these endogenous pyrogen is interleukin 1, and other cytokines released by the macrophages such as a tumor necrosis factor, they will also produce fever. But the net effect is a septic shock. So when a endotoxin, especially lipopolysaccharide has been produced, you get a fever inducing substances and it leads us into a septic shock. Now, certain gram-positive bacteria as well as pathogenic fungi could also lead on to septic shocks, but most predominantly it is the endotoxins which mainly lead on to the septic shock. Now, what is septic shock? Okay, the septic shock is a situation where what happens is it activates a protein called the Hagman factor, the clotting factor 12, and it overstimulates the immune system. And so what happens, large amount of factors which have been immunological factors gets activated, and that would result in the what multi-organ failure. I'm not going to the details more precisely, but do have an understanding that both the exotoxins as well as the endotoxins do have an important role in the virulence of the organisms. When exotoxins serve as superantigens in some cases and causes multi-organ failure, the endotoxins would serve as what? They could lead us into septic shock because of the presence of various immunological factors which have been triggered, or pyrogens which get triggered by the presence of the endotoxins. If you move on, you can see that various plasmids do play an important role in the virulence of the organism. As we had seen earlier, various virulence factors were being coded by the plasmids. And see, so those plasmids which code for the virulence of the organism, they are referred to as a virulence plasmids. Now, every different organism would have a different name for the plasmid which is causing for its virulence. So, I am not going into the details of that. But in addition to these virulence plasmids, we could also find that various multiple drug resistance encoding plasmids also do occur in the case of pathogens, and that could lead into the virulence of the organism. Similarly, we have also got bacteriophages, which induces virulence. Now, for example, in diphtheria bacilli or in Corynebacterium diphtheria, which causes diphtheria, you can see that the gene for the toxin production has been present in the tox plus corynephages. There are various other examples also, as we had mentioned earlier. So, the presence of bacteriophages associated with the bacteria could sometimes contribute towards its virulence. The next factor which influences the virulence is the communicability of a particular disease. Now, what do you mean by communicability? It mainly refers towards the ability of a pathogen or a parasite to spread from one host to the other. And it does not affect the prediction of the disease, but affects the survival and the distribution of the parasite or the pathogen in a particular community. And you can see that virulence and communicability need not to be correlated, because generally if you go to look, pathogens, they shed in secretions as in respiratory and intestinal diseases, and they have high communicability. Communicability means the ability to spread from one host to the other. Of course, you can recollect the difference between invasion. Okay, invasion is what? What? You do remember what is invasion. Invasion is something where you invade a particular cell, or you what? You enter into the cell. Okay, but this is not into one the cell we are talking about in the case of communicability. In the case of communicability, we talk about when the organism moves from one host to the next host. In invasion, we are saying the ability of the pathogen to spread from one tissue of the particular cell to the next tissue. But in the case of communicability, it is moving from one host to the other. And you can see that some human infections represents a dead end. As now in the case of rabies or hydrophobia, it is a dead end. Human is considered to be as a dead end for the rabies virus. And for pandemic and epidemic diseases, it mainly requires strains of a pathogen which possess high degree of pathogenicity as well as communicability. So the ability to spread from one host to the other mainly refers towards a communicability. And other than the toxins, various other bacterial products also contribute towards its virulence. Like you can have certain cases in which high alluronidases, these are enzymes which mainly split high alluronic acid. It is a it's a main component of the intercellular connective tissue, and thus it facilitates the spread of the infection along the tissue spaces. You can have leukocoordins which damage the polymorphonucleoleucocytes, or the WBC is being inhibited. So as a result, what happens, or the as a result, the immunity of the person gets reduced. And sometimes many pathogens produce hemolysins, which are capable of destroying the RBC. But their significance in pathogenicity is not clear exactly. And there are certain other organisms like staphylococci, which will produce thrombin-like enzyme coagulases, which would prevent phagocytosis by forming fibrin barrier around the bacteria and walling off the lesions which have been caused by the pathogen. You can also find bacterial appendages now in the case of Klebsiella pneumonia as well as Haemophilus influenza. They also do have capsulated capsules which will prevent them from easily getting phagocytized. And in the case of Salmonella typhi, Klebsiella and all that, as well as in the S typhi as well as the K antigens of E. coli, not Klebsiella, they will help to withstand phagocytosis as well as the lytic activity which is being caused by the complements in the case of the bacterial surface antigens. So the various bacterial appendages which have been found to be associated with the bacteria would also aid to prevent it from getting phagocytosed by the bacterial cell. By the host cell. Now, in addition to this, you can see that the infecting dose or the amount of bacteria or the number of bacteria which happen to enter into the cell also plays an important role in its virulence. You have mainly two terms here. You have the minimum infecting dose or the minimum lethal dose. As the term implies, minimum infecting dose is a minimum number of bacteria which is mainly required to reduce a clinical evidence of a particular infection. Whereas a minimum lethal dose is a minimum number of bacteria which has been required to produce the death of the host. And since when we consider there would be different variations, we normally like to make it more statistically significant, we use the term infectious dose 50 and lethal dose 50, which mainly represents the number of organisms or dose that will infect or kill, respectively, 50% of an experimental group of hosts within a specified time period. I guess the significance of infecting dose is quite clear to you, because only when you get infected by an organism to a particular level, or when large number of bacteria happen to grow within you only, you might show a clinical disease. When they are being found to be in large amount, in a very minimum or a small amount, you might not show a clinical disease. And so the minimum dose of the bacteria or the minimum number of bacteria which will give you the clinical evidence of infection is referred to as a MID. And the minimum dose of the bacteria which will cause death of the particular host is referred to as a MID. And to make it more statistically significant, we have the ID50 and the LD50 to be mentioned. So these are some of the examples of the virulence factors which mainly contribute to the organism's virulence. Hope so it is clear to you. With this, we come to an end of the virulence factors which mainly contribute to the virulence of the organisms. Thank you for now.